second year biochemistry, and I was teaching something difficult like Sanger sequencing or next generation sequencing, and he came to me at the end of the lecture and asked me a question, and I didn't know at this point, and the question went on, and it went on, and it went on. And I had to stop him so that we could walk out of the room so the next lecture could start. But he sort of said something that, could he do a project in the lab or something like that? And I said, oh, whatever, just send me an email. And which he did, of course. And I read it and thought, oh, well, maybe I should do something. And then ping, you haven't answered my email. And so I actually got a very clear insight into Yiheng just from those initial meetings. He's a very determined and persuasive individual, particularly if he's looking to get his receipts refunded. And as you also see during his talk, he doesn't let things get in his way. He overcomes problems as they come. He takes on difficult problems and tries to solve them. And anyway, he can explain that to you during the course of his seminar. So from there, I think you did a undergraduate thing with me, right?
Master.
No, before that.
Yeah, that was a research course, master course.
See how long we had this up. And then he did the masters, which is the honours here, and he got a first class for that, working with Ben Schlesinger here on strike rust. And after that, he wanted to stay and we wanted to keep him, but we weren't sure what to do. Because being an international student, it's quite hard to get money. But then we were lucky that Eric Stone from BDSI had some money. And so from there, we had to come up with a project. And what you'll see is it's a very modern project, a very 21st century project, where there's a ton of supervisors, including myself, Ben here, Eric himself, and really significant input from people like Andrew Milgate at Wagga Wagga. And what he's done is to take really cutting edge technology and apply that to a real world problem, which is cropping, and to ask in those crops what microbes are present. Can we identify the pathogens are there? Can we take that a step further and identify what other organisms are there as well? So once again, he's going to explain that to you, and I won't. Now, let's talk about him on a personal level. So, very large personality. He's very curious. He's often asking curious questions in seminars. He's very big on culture and politics, and we've had great discussions in the lab. India versus China, and I think China won. I certainly know that China didn't back down. Apart from that, he's had a short career as an avid car collector, several cars at one point. And he's made an impact on Australia in that there are less sea urchins here than there were when he arrived. The PhD has gone extremely well. He's published 3 papers. There's another one in preparation, and he's got a really first-rate postdoc position in Germany, in Tübingen, with Eric Kamen, who's one of the really exciting young researchers coming through in microbiome studies in plants. So with that, I would let him go, but now I want to let Ben get his 50 cents in as well.
So I'm the second supervisor. I think Eric is actually not here. Maybe he's online. So Yiheng wanted to say me something. Wanted me to say something too, I think. I only said good things. So when he initially came, he really tried to game the system, and I think that was a little bit like his kind of attitude, kind of not really aspiring to higher intellectual academic goals, but really like, how can I get the best grade? Which is also something. But what I really appreciated working with Yiheng over this year is like seeing him mature as a scientist. I think it was really excellent. Where he initially had this gaming attitude, he really got into it and really like read a lot of papers. And I think it was especially inspiring because he actually didn't have biology as an undergrad, but chemistry. So I think that was actually quite nice to see that at the end, especially now, you will see some of it is really biological question. And I hope actually moving on to Eric's chemistry lab in Germany, which is actually just the word for German, I can recommend. I think he is really set up to hopefully do very well. We hopefully we see him back sometime. I'm not going to say anything else.
Thank you so much. This is too much for introduction, and good afternoon. Thank you. Good afternoon. Thank you all for coming. It's my pleasure today to share with you my PhD project about pathogen detection and the microbiome analysis with the focus on fungus. I'd like to Where's the all right. Oh yeah, there you go. Thank you. All right. So I'd like to start from this top 10 unanswered questions from the most recent Congress of Molecular Plant Microbe Interactions in 2019, which is the most recognized Congress in our research field. And as you can see, there are two highly voted questions were either about this beneficial microorganisms or the microbe-microbe interactions. So I think this indicated that now scientists have more and more overviewed the plant microbe interactions in a more complex manner and included many of these small players on the table. And the research on these small players has become more and more hot topics. So to summarize these complex interactions, it comes to this very well-known disease triangle. So since we got some time today, I'd like to give you a detailed introduction about this triangle before digging into all this bioinformatics. So generally, as you can see, three components: plant host, pathogens, including fungi, bacteria, viruses, etc., and also the environmental factors, including abiotic factors such as heat, drought, cold, and so on, and also the biotic factors like the microbial. So my PhD is primarily focusing on the fungal diseases and their associated microbial communities. So for today's talk, I would like to simplify this triangle to be the plant host, fungal pathogens, and the microbiome. So I think the first and I think the most well-studied interaction within this triangle is between the plant host and fungal pathogens. So here in the image is a typical example showing you a wheat plant from the field infected by various of fungal pathogens. And these fungal pathogens are actually responsible for the majority of the yield loss for most of the crops. And in wheat, it's more than 90%, which is a very important problem. So the molecular basis of this plant pathogen interaction actually has been formed decades ago and can be nicely represented by this simplified figure. So generally speaking, there are two layers of interactions or two layers of immune responses, namely PTI or PAM-triggered immunity and ETI or the effector-triggered immunity. So because my study is not really about this, so I don't want to talk too much on these detailed processes, but I just want to say that there has been a lot of study going on in this area from RSP and leading to the identification or like discovery of many these immune receptors or immune receptor genes be used in our crop varieties to help them fight against pathogens. So moving on, the next interaction I want to introduce is between the plant host and the microbiome. So actually this area is also historically very well studied for the symbiosis process between the plant and the rhizobia. However, in recent years, people more and more revealed that this plant microbiome is rather complex than just doing symbiosis. So generally now we group the plant microbiome into these four categories based on their different origins. As you can see in this figure, the above plant surface microbiome is named the phyllosphere microbiome, and the root surface microbiome named the rhizosphere microbiome. Also, the endophytes within the plant tissue is named the endosphere microbiome, plus this bulk soil microbiome from the environment. So obviously, the different microbiome will have contain different species. So it is important to define the structure by describing what kind of species are present and how much abundance of each species. So here I'm showing you a typical description of the microbiome structure, which is a principal coordinate analysis that show the microbiome can be basically grouped into based on their different geographical locations, based on this like shown by this top and bottom panel, and also can be divided by the left and right panel based on their different sample types, such as this endophytes and the soil or rhizosphere samples from this left and right panel. So eventually this kind of microbiome structure is to help us understand the function of the microbiome, and also to somehow utilize the microbiome to help the plant to help the plant growth or help them to be more resistant to pathogens. So here I'm just showing you one typical example under the context of this disease triangle that they proposed to, this idea proposed to utilize the certain bacteria to artificially synthesize the different bacteria strains from either rhizosphere microbiome or soil microbiome, and into this artificial bacterial community to help the plant be more resistant to pathogens, or in other words, to help the plant to create a disease-resistant phenotype without changing the genetic background of the plant. So, okay, so that's the general overview for this plant microbiome interaction. So moving forward, the last interaction is between the microbiome and fungal pathogens. So here, actually, I would like to take a step back first and talk a bit more about this special kingdom of fungi. So fungi, in my opinion, is a very low-key kingdom of organisms, which they actually played very important roles, but were not much noticed. So here is some representative species from different classes of fungi. And as you can see, for example, some of them can be food just by themselves, such as this oyster mushrooms. And also this one is white jelly fungus. I'm not sure if you have tried it before, but very popular in China. And and also there are two types of yeasts, which are important components for both baking and brewing. And on the other hand, fungi can be important plant pathogens, such as this Crohn's smut disease caused by Eustrago maydis, and also this penicillium species, which is a causative agent of mold. So the fungi and the microbiome could also form different types of interactions, such as the symbiosis and competition. And actually, this study also started since long back. And for example, you can see this paper published in 2005, showing a plant pathogenic fungus can harbor this symbiotic bacteria to help them produce toxins and to better attack plants. So the competition relationship, I think the example has been many and can be represented by a lot of these studies about the biocontrol agents from different fungal diseases, such as this commercialized product trichoderma. So although, I think this kind of microbiome fungal pathogen interaction is important, but I think it's still at the moment the most poorly understood interaction within this disease triangle. And I think the reason for that is mostly due to the challenge in characterizing fungi. And there are several reasons for that. So first, most of the time we will need the microscope to study fungi simply because they are too small. And also many of the fungal species, including a lot of important pathogens, are not really culturable. So you can't really separate them from their host. And secondly, a lot of fungal species have unique and complicated life cycles. So because our lab studied wheat-stripe rust, so I'm showing you the life cycle of it as an example. So as you can see, all of these images come from the same wheat-stripe rust fungus, but just on different stages of their life and on different hosts. So you can imagine that if you characterize them only based on the morphology, they all will be easily grouped into completely different species. So, and this point actually, I think, leads to the most important reason for the challenge, which is the complex genomes of fungal kingdom and their very poor status in the reference genome database. So I think the fungal kingdom is relatively like a large kingdom of organisms, have I think around 5 million species in total, similar to animal and plants. But if you look at the proportion of the reference genome database, it is very small. The fungal proportion is very small and often not much organized. And I will show you the data to support this. So under this conceptual network, I built up my overall topic of my PhD is about fungal classification using the sequencing technologies, sequencing strategies. And I set up 2 objectives. So first is to ask if we can detect those fungal pathogens and briefly describe their associated microbial communities through this metagenomics approach. So just in case if you are not familiar with the term metagenomics, it's basically a study of the total genetic material or total DNA directly recovered from environmental sample. And so it will contain all the species together, and we sequence them together and decompose them using different bioinformatics tools. So the second objective is to see if we can further improve those fungal detection or broadly speaking, species identification, and also the subsequent community composition analysis. So basically to see if we could also improve the calculation of relative abundance for each species. So for today's talk, I would first introduce you 2 proof of concept studies about the pathogen detection, and then I will mainly introduce you a method development project using a mock fungal community to try to improve the fungal classification further. So for the first wheat pathogen detection project, we collaborate with Dr. Andrew Milgate's group in Wagga Wagga Agriculture Institute. And as you can see from this map on the left, the green area indicated the major wheat growing region in the eastern side of Australia, or as we know, the wheat belt. So you can see that Wagga Wagga is located right in the middle of it. And over there, they have different disease nurseries and testing different wheat varieties. So as shown in this image, we also have a small disease trial. And so you can see each of this sign indicated one specific wheat variety that has been tested for one or multiple pathogens. So the overall goal of this project is try to track the development of different fungal pathogens and their associated microbiome during multi-year infection cycles. So here is the general workflow for the pathogen detection. And as you can see, generally consists 3 steps. So first, our pathologist collaborators will go to the field, take the leaf samples with confirmed disease phenotypes, as well as some healthy wheat leaves as a negative control, and send to our laboratory. So in the lab, I did a blind DNA extraction. So I don't know what is in each sample, and follow this standard procedure to prepare the sequencing library, use this DNA barcode to label each sample and put them all together for one sequencing run. So in the lab, I used this portable MinION sequencer, generated the nanopore long reads for the bioinformatics analysis. And also in the lab, I developed a data analysis pipeline, including 2 BLAST steps to try to identify all the major pathogen species, and also to describe to see if we could describe what other species are present in each sample. So I first blast all the reads against a specific reference database from the genome of 5 species, including wheat, and 4 pathogen species responsible for 4 different wheat diseases. So 3 of them, the first 3 of the pathogen species that we know present in sample confirmed by the pathologists. And this particular one, Parastegonospora nudorum, is previously known not prevalent in our sampling area. So I put its genome in as a negative control for the database. And we don't expect to see it in the result. So one example result is shown in this figure. As you can see, all the striking bars represented the total amount of reads identified as different pathogen species encoded by different color. Typically, the barcode in the x-axis is basically just different samples. All right. So overall, all the result from the sequencer actually matched to the phenotype or confirmed by the pathologist. Especially you can see this healthy read control. There's no read from this sample was identified to any of the pathogen species. However, we also find some certain limitations from this shotgun metagenomics approach. And first is that, as you can see on the top right corner, the wheat sequence comprised overwhelming percentage of the data. So in this sample, it's about 90%. So kind of about 90% of data is kind of unused. And also, we found that this approach might be a little bit problematic in the identification of double infections. For example, in this barcode 4 sample, supposed to be a double infection by both strep rust and Septoria triticide blotch, but you can see the reads identified as strep rust was not very significant. However, again, this approach also offered us a very nice surprise, actually. So you can see, as you can see in the barcode 2 sample, there is a reasonably significant signal identified from this Pinodorm, Parastagonodorm, which, as I just mentioned, is supposed to be a negative control species, and we don't expect to see it in the result. So what happened is that eventually I have to dig out to the fridge and dig out all the DNA samples from the freezer. And we did a PCR test using the Pinodorm specific primer, and actually found that this species is present only in the barcode 2 sample, but not in the others. So this result actually suggested that this shotgun metagenomics approach is feasible for might be feasible for the identification of some unknown species. So now I've shown that the identification of all the major pathogen species. So the next question is to see what other species are present in each sample. So I blasted all the remaining reads against the NCBI nucleotide database, which is supposed to contain all the sequenced genomes. And the result of the top 5 genera identified in each sample is shown in this figure. So as you can see, from all the samples encoded by different color, the relative abundance of each genera of species are all different in each sample. So this suggesting that the microbiome profile is pathogen specific. And the relative abundance of all the different samples are all increased actually compared to the healthy weed control, the microbiome of healthy weed control labeled encoded by the purple color. And you can see that only a small proportion of weeds identified as Pseudomonas and nothing else. So very interestingly, early last year, there was a paper showing the same finding as we did on the Septoria triticide blotch infected samples, that both of us found that the population of relative abundance of Pseudomonas significantly improved along with the Septoria triticide blotch infection. So just want to say that although they didn't cite us, I'm quite happy about this result, because it means that our result is most likely true. But just want to clarify that in case if you see somebody else complaining on Twitter, that's not my opinion. Anyways, just some quick summary here. First is that we can detect fungal weed pathogens from field samples using this shotgun metagenomics approach. And I also found that the microbiome profile are pathogen specific. However, we also identified certain limitations from this shotgun metagenomics approach. So we decided to move on and have more trials about this strategy. And so at that time, there was a great opportunity to also collaborate with Professor Meyer's group at Sydney University to apply this method on the application of certain human fungal pathogens. So this time the pathogen is called Pneumocystis jirovecii, causing pneumonia and often found in the immunocompromised patients. So it's also not culturable like the many other fungus, and traditionally very time consuming to diagnose. So you can imagine that once got the infection, it will be a very dangerous disease. So we set up this pipeline for diagnosis. So this time we got 3 patient samples with confirmed pneumocystis infection, as well as 3 healthy individuals as negative control. So thanks to our collaborators, they extracted DNA and sent us for sequencing and analysis. So this time I tried two different analysis pipeline. So also used, first is also this alignment blast approach, and also this K-mer-based analysis supported by the nanopore company, which is named called WIMP, or short for What's in My Pot, basically. And the K-mer analysis classification, the principle of this K-mer classification is to use this this fixed length sequence as a signature, and to build the signature from the reads and compare the signature that was built originally from the reference genome database. So in this way, it will become much faster compared to directly align the whole read to the reference genome. So here I'm showing you the blast result of the top 18 genera of species identified in each sample. And clearly you can see that the pneumocystis only identified it from the patient samples and not from the healthy individuals. And on the left, I'm just showing you the total amount of fungal reads that was obtained in each sample to give you an idea, like how much would give you an idea of the quantity of data that is needed. And clearly we don't need much data to achieve this result. So in here, we didn't really dig too much into the microbial diversity here because we mostly focus on the diagnosis. But I think this result also proved the concept that this shotgun metagenomics approach is feasible for the identification of at least the pneumocystis infection from the human clinical samples. However, we also find a major, actually I think a major problem in specifically for the fungal identification from these human data sets. And here is the example. So here I'm showing you bacterial identification first from one particular patient samples using these two approaches, as I just mentioned, the alignment on the left and the K-mer classification on the right. So as you can see, the length of the vertical line here indicated the total amount of reads identified based on these two approaches. And the green area indicated the reads that are mutually identified as bacteria by both approaches. And you can see this very thin pink line here indicated the reads identified as bacteria by K-mer approach, but the non-bacterial or unclassified by alignment approach, and this thin blue area is just the opposite. So from this figure, you can shows that the bacterial classification is largely consistent between these two approaches. As based on the length of these vertical lines, they classify similar amount of reads as bacteria, and most of the reads are actually mutually agreed as bacteria. However, if you look at the fungal classification, so you can see these two approaches are classified so different amount of reads. First, so different amount of reads as fungi. So alignment only classified very few reads as fungi, and K-mer classified much more of the reads as fungi. And also, you can barely see the mutually agreed reads as fungi between these two approaches. So this result actually suggested that no matter what kind of pipeline used for fungal classification, it is the fungal data themselves that is difficult to be classified. So based on this problem, we further designed this method development project to try to improve the fungal classification by benchmarking different classification strategies on mock fungal community. So again, thanks to our collaborators, made this great mock fungal community, I believe from 46 different fungal species, mostly human fungal pathogens. And we put them different ways. So we first put them with same amount of DNA, and we also put them with same amount of tissue before the DNA extraction. So we used different sequencing strategies, the shotgun metagenomic sequencing, and also the amplicon sequencing strategy with both nanopore long reads and Illumina short reads techniques. So in total, we have 4 data sets each for the 2 samples. So the overall aim is to first try to identify what is the best method for the classification of fungi for each data set? Or in other words, what is the best combination of algorithm and database for the fungal for species classifications? And also, subsequently, we want to see if we could further optimize the community composition analysis to see if we can also improve the calculation of relative abundance for each species. So given time limit today, I would just only focus on the data sets from the shotgun metagenomics approach, but I would be more than happy to share with you the result from Amplicon data later, if you are interested. So just a quick note here. So for all the nanopore reads, because they are all long reads, so we directly use the reads themselves for all the classification analysis. But for the Illumina data, since they are all short reads, so we first assemble them into contigs and use all the contigs for the classification. So in total, I tested 2 algorithms and a combination of 2 algorithms and 2 databases, the K-mer-based algorithm with Kraken, and the alignment-based algorithm using BLAST. So the two database is NCBI nucleotide database and also another more specific fungal database from Rathjen. So to evaluate each classification, we first checked their accuracy, and then I also tried to tweak the parameters around to see if we could further improve the accuracy. So in order to check the accuracy of the classification, I used these 2 parameters, recall and precision. So recall is basically the percentage of data that classified data, and the precision is the percentage of identified reads that belongs to the species in the mock community. So you can imagine that the precision is a direct reflection of the accuracy of the classification. So we want the precision rate to be as high as possible. So here I plotted all the recall and precision for all these different classifications from all the data sets, and it's 2 samples. Left is poor DNA, right is the mixed culture sample. And as you can see that no matter, and as you can see that all these filled green dots have resulted in the highest precision, which is the x-axis, always resulted in the highest precision. So all these 4 classifications are actually from the same combination of algorithm and database, which is the BLAST algorithm against the specific fungal database. So this result indicated that the best classification or the most accurate classification is using this alignment-based algorithm and a more specific database. So now we know what is the best classification. And so the next question I ask further is to see if we can further tweak around the parameters to further improve this best classification. So actually this comes to a little bit a tricky point about how we actually evaluate the alignment. So here I'm showing you a typical alignment case. As you can see, the query sequence on the top and the reference sequence at the bottom. And generally you will have some, typical alignment, you will have some mismatches and you will have some gaps in the alignment. And all these different alignment algorithm will give you some quality measures such as the e-value, percentage of identical matches, bit score to evaluate the alignment based on the quality. So here is the example of the parameters, which is the percentage of identical match using the length of the blue area divided by the length of the red area. However, I think that this kind of quality measure cannot always represent all the alignment cases, especially when you think about a very long query sequence. So sometimes you will have a very good alignment, but only from a very small proportion of the read. So this time, in this case, the alignment quality will be really high, but it may not be the most accurate alignment because only comes from a very small proportion. So I come up with this new parameter called the alignment proportion, and which is basically the proportion of the read that was identically aligned to the reference genome database. And in this case, I think it will also consider not only the quality of alignment, but also the quantity of the alignment based on the length of the query sequence. And in this case, we think it will be a better parameter to evaluate the alignment. And so now I'm showing you the data to support this argument. So what I did here is that I applied cutoff on all these different parameters, like the e-value, percentage of identical matches, read length, read quality, as well as this alignment proportion parameter. And what I found is that applying cutoff on, so I applied cutoff on all these parameters from the result of the best classification, as you remember, the best combination blast algorithm against the fungal database. And so I found that applying cutoff on the alignment proportion can improve the classification the most. So as shown in this figure, the x-axis is the cutoff on the alignment proportion. So 20% means that at least I need 20% of the reads to be identically aligned to the reference database. And all these different colored lines indicated the change of different aspect of the classification. So the red line is the change of the precision rate, and the green line is the change of the completeness, which is the proportion of the number of species identified from the mock community. So obviously we want all the species to be identified in the mock community, so we want the completeness to maintain 100%. So, and the green, and the blue line is basically to tell you how much data left after the cutoff. So we name it as remaining rate. So as you can see here, as long as you apply a very small cutoff, like 10% or 15% on the alignment proportion, the precision can improve from roughly about 95% to nearly 98%. And the completeness always maintain 100%, and the remaining rate is roughly about 90%, which is acceptable. So then we look at all the others. So this is only the data from one sample. So, and then we further look at all the other samples and actually found that all the other data sets have a better improvement after applying cutoff on this alignment proportion compared to this the sample I just showed, which is a nanopore data from the pooled DNA sample. And especially you can see this Illumina data from this mixed culture sample, the precision rate improved from 91% to nearly 99%. And the completeness always maintained 100%. The remaining rate is also always acceptable. So I plot out this figure for all the other parameters, the e-value, read length, read quality, and I found that this parameter, this alignment proportion parameter, is the best. So here is just the example to show you another parameter that didn't work very well. So this is a cutoff on the read length in the x-axis in base pairs. And as you can see, the precision, the red line, didn't improve at all, or sometimes even dropped. And also this completeness also start to drop in a very low cutoff level. Okay, so now, so far I have shown you that the best classification is from the alignment algorithm and the specific database, and also applying cutoff on the alignment proportion can further improve this result. So the next thing I'm going to do is to have some water, because the next few slides will be a bit tricky. Thanks. Okay. So I've showed you the improvement of the classification. So, and the next task is to deal with the community composition analysis. All right. So the idea here is for the community composition analysis, or the calculation of relative abundance for each species. The idea here is to generate a gold standard analysis and compare all the other community compositions from other classifications, other different combinations of algorithms and databases with this gold standard, and see which one is the best. And so to construct this gold standard analysis, I first generated most restricted database, because we just know that the most specific database is the better identification. So I generated the most restricted database from only the genome of species within the mold community, because we know what we put in. So you can imagine that in this case, every single read, as long as it's classified, it will belong to the species in the community. So in this case, the precision rate will be 100% all the time. And also in this process, we sequenced and assembled 3 new genomes from the species in the mold community, which doesn't have reference genome in the database. And as you can see, we only use the nanopore data for the assembly, and the result is pretty good based on the number of contacts and the Busco ratios. So after the construction of this most restricted database, I used the alignment algorithm to map all the reads back to this database and generate the gold standard classification, and then subsequently calculated the gold standard community composition. So basically, a gold standard analysis will include a gold standard classification and a gold standard community composition. So then I compared all these different combination of algorithm database. I calculated the community composition from all the previous analysis and compared those community composition with the gold standard. And the idea is that if one particular community composition is more similar to the gold standard community composition, that combination of algorithm and database will consider the best for the community composition analysis. All right. Anyways, so lastly, again, I applied cutoff on the alignment proportion to see if, so after identification of this best method for community composition, I also applied the cutoff on the alignment proportions again to see if the similarity can be further improved. So to quantify this similarity, I used this statistical measure called the Bhattacharya distance. So just in case if you're not familiar with statistics, it's basically you can basically imagine it as an angle. So if the angle is smaller, meaning the 2 community compositions are more similar to each other. And if the angle is larger, means the 2 community compositions are more different with each other. So, and the result, so I calculated the community composition from all the previous classifications and compared them with the gold standard community composition. And what I found is that, again, the same as previously, the BLAST alignment method against the specific fungal database resulted in the closest Bhattacharya distance with the gold standard community composition, meaning that this combination for this classification is also the best for the community composition analysis. So just to show you, like, visualize how different they are. So this between this gold standard and this best practice. Here I plotted out the relative abundance of all the different genera of species from both classifications. And as you can see, there are indeed some inconsistency here, but I would say the overall trend are still similar. So lastly, again, I applied cutoff on the alignment proportion and recalculated the community compositions from after the cutoffs, and recalculated calculated the change of the Bhattacharya distance after the cutoff on alignment proportion. And as you can see that in all these 4 data sets, all the Bhattacharya distance all decreased compared to the original point, which means that they become more similar to the gold standard. And this result suggested that applying cutoff on the alignment proportion can further improve the community composition analysis. So there's another small problem is that this Bhattacharya distance is not very commonly used. We found the Bhattacharya distance not very commonly used for the measurement of similarity between different community compositions. So for the verification of this result, we used another statistical measure called the Euclidean distance and did exactly the same experiment again. So Euclidean distance is found more in the previous literature for the assessment of community compositions. And it's basically, you can imagine it as absolute distance within between 2 points in the multi-dimensional space. So again, it's also like the smaller the distance, the better, the more similar the two community compositions. Okay, so I did exactly the same thing, just replaced the Bhattacharya distance with the Euclidean distance. And as you can see in the mixed culture sample, which is the round dots, it also shows a decrease after applying cutoff on the alignment proportion. However, in the pooled DNA sample, it actually, you can see the Euclidean distance actually increased. So which is kind of like a controversy result with compared to previous. So then I dig further into the statistic formulas of this Euclidean distance, and eventually found that it's actually not the similarity decreased, but it's more like because the relative abundance of one particular dominant species from this pooled DNA sample. And the change of the relative abundance of that particular genus was too influential for the calculation of Euclidean distance. Oh, okay, I won't be elaborating too much here, but we can, if you are interested, we can talk afterwards. But overall, this still supports the argument that applying cutoff on alignment proportion can further improve the community composition analysis, and also suggested that the Bhattacharya distance is a better measurement for the similarity between the community compositions than the Euclidean distance. So that's pretty much it. Just to recapture all the key messages again. So first, I've shown that we can detect all the fungal pathogens and describe their associated microbiomes using this metagenomics approach. So putting this into a bigger picture, I think it is very promising to integrate this approach into the future disease surveillance framework. So last year we wrote a review, summarized all the biotechnologies used for the help protect the plant biosecurity. So, and along with many others, as shown here, you can see the sequence, and as we think the sequencing-based approach is have a great potential to be used for both point inspections, such as at this customs for importing and exporting inspections, but also this large-scale field disease surveillance system for different crops. So secondly, I showed you the most accurate classification is between the alignment algorithm and the specific database, and also applying cutoff on the alignment proportion can further improve the classification and also the community composition analysis. So you may definitely ask that what is the significance of this, this finding and what it can be used for. So here I just want to revisit these top 10 unanswered questions again. And for example, here in the question 4, to investigate how do microbe interactions affect the plant microbe interactions. We first need to characterize those microbe interactions. And several important studies have already shown that a better profiling or better characterization of the microbiome or the general microbial community can lead to significant improvement on the detection of microbe interactions. And this improved detection of microbe interactions can obviously provide us more insights on these important unanswered questions. So that's it. Thanks to all the people who helped me, especially my supervisors. And here I just want to say that I have an amazing supervisory panel, and each one of them played an unreplaceable role for my PhD. And I couldn't even imagine to even start my PhD without any one of them. And also, second, I want to thank all the collaborators. It's been my great pleasure to work with you guys, and thank you. I really appreciate your help, your patience, and also your trust. And next, I want to thank to all the lab members from past and to the present, and also broadly speaking, the whole MPMI community from both RSP and the CSIRO, which I think provide a fantastic environment for me as a PhD student to learn all these cutting edge sciences. And also I want to thank to the plant service team and the IT service team and also other RSB supporting staffs for the help and who bothered by me millions of times and being so patient all the time. And lastly, just a special thanks to all the family members and friends supporting me, especially Yuzhou, who is my wife now, and have been not only very doing a great job in her own PhD, but being also pretty supportive for my PhD journey. And also thank you for listening. I'm happy to take any questions. Thank you.
Disease, when you have samples that already show a lot of disease, do you think you could also use it to find disease that is not visible yet in the field? Or
so I think as I showed for the detection of Pinot I think it definitely has this potential, but I think we do need to some study to define the threshold for the sensitivity of it. Definitely, this is a good question.
yeah, and how quick is the turnaround time for all this, like from getting the sample to getting your result?
So it depends on how hard you push the edge, but there was some point that I've been thinking of using this portable sequencer just directly in the field, and however, at that time it wasn't that achievable due to many factors, such as like the DNA extraction, and also the purification of DNA and the library preparation time. But I think now I've seen many studies doing, especially in the human detection of human pathogens, they can turn around in half a day, 6 hours, something like that. Oh.
Hey, Yiheng, congrats first. So I have a simple question. When you were dealing with sample mixed of like different type of fungus, how do you deal with the conserved or common sequence between them?
So that's a really good question, actually. So generally, the method I used is most of the time is alignment method. So theoretically it should be accurate. So tend to basically, even if it's conserved sequence, it tends to classify the read into correct species. However, I think even if it's not, I think the bias is most likely average. So it's kind of no influence, hopefully no influence, but indeed I haven't tested that for specific regions.
Yeah, so do you think that's the reason why the alignment method would be better than the other one?
So I think both have pros and cons. So actually what happened, I also tried the alignment method for the amplicon data sets. So amplicon data is from conserved regions. And so the problem for, say, for example, if you want to blast amplicon data set, it will take forever, even with a very small data set, like 10,000 reads, something like that. But the camera will give you answer within like 10 seconds. So this is, so as I would say both have pros and cons, but definitely alignment is more accurate if you only look for the accuracy. So, and by the way, just want to extend this a little bit. So I think there are studies only focus on the, the protein coding genes or like non-conserved regions, only build database based on that. And I think Diane Sanders' group in the UK, they did a lot of work on this amplification of this region and build database for that. Uli?
Thank
you. Thank you.
Do you know if there's anything known about whether a plant that gets infected by a pathogen can assemble a protective microbiome?
So plants get infected.
Does the plant recruit certain other bacteria?
Yeah. So this is a really good question. And actually, what happened is that my future, like my postdoc will mostly, I think my postdoc will mostly focus on this direction. So what happened now is that a lot of studies has been focusing on the questions around how this, how the plant actually interact with this microbiome and how this specific microbiome was formed. And the most, I think some argument was saying that they function in a specific small group of taxa and use that small group taxa to guide to interact with all the remaining microbiome to form a specific protective microbiome. So I think that's one of the hypotheses. However, one of the important problem or issue might be in this process is how stable this interaction will be. So, so it's, I think the first question we need to investigate further is to actually look for stability of the microbiome, or on the other hand, the dynamics of the microbiome, how they actually change and form stable forms after different stimuli, like after different infections, like pathogen infection or even abiotic factors, something. Yeah. Not sure if that answered your questions.
So we have two questions online, sorry, 1 minute. So the first question from Gupta is asking, what is your experience with the depth of sequencing required for using the metagenomics approach to pathogen detection?
That's also a good question. And so I don't have really much experience because I only deal with a few data sets. But for my wheat pathogen detection data set, I think the coverage for the wheat strep rust is around the 0.2-fold. So it's actually quite, I would say it's reasonably sensitive. And you can, with about 0.2-fold of genome coverage, you can basically get a pretty sure answer for the detection. But it obviously will depends on like the genome size of the different pathogens.
So the second question from Tim Hewitt asking, how many mix of infected material did you use for the MinION sequencing, and what minimum sequencing depth was required for the identification?
Yeah, I think it's similar with the previous one, but depends on different different samples. And so for human samples, I think we got the answer from very little depth of the sequence, because I think for the human sample is also mostly comprised by the human reads. But for the wheat samples, the sequencing depth is, I think, so if you also include the host, it will be quite high. But so now the one of the direction is to see if we could actually isolate the host DNA, like deplete the host DNA and only sequence the microbes. And if that's the case, then we only need a very few depths. Oh no!
Don't worry, it's an easy one. So you talked about in your samples you saw fungi and bacteria, but there's representatives from other kingdoms I would have expected to see that weren't there, like marsupials, nematodes, protists, these sorts of things, viruses. None of those signals in your data sets?
Honestly speaking, I haven't looked too much on that part. And also, I think the number of samples that we had is also quite limited. And I think the other, somehow, I think we found some nematodes from the human fungal, like human clinical data set. So I mean, obviously, this will suggest that it will have give you like a comprehensive view of all the potential like infections or all the potential diseases for each sample. But again, it also brought the biases for the like for the classification. So certainly we will need to further improve the accuracy and to be more confident on all this effect, like all this result. So that's why we have this method. Sorry? No, the answer is we had some other species. We definitely observed, but just in terms of the confidence.
Did you use it from extraction to sequencing?
Yeah, so we have this mock fungal community so generated from the individual species that from our collaborators. And they extract DNA individually, or they pull the tissue together, equal amount of tissue together for the DNA extraction all together. And so basically we have two, I would say, biological replicants. Yeah. So.
Seems like Ben already indicated he don't want to ask any questions.
No.
No, actually, just to say thank you to Yiheng.
Oh, thank you so much.
Just to finish it off, I think you could really appreciate like the scholarship he actually developed over the years and the depth coming from chemistry, and then actually asking a John silly question about Sanger sequencing in his lectures to now. And I think you also could probably appreciate that he actually was really collaborative and really established excellent collaboration with Wieland Meyer and Andrew Milgate, which we will profit from for years to come. And I think the last thing which didn't come through, which was really hard to actually do in a presentation, is that he's a great colleague. I mean, he was really great to help other people in the lab, and he also got a couple of publications out of this, and other master's students, honor students, I think would really appreciate it. So
Thank you so much.
I advised Yiheng for his master's when he first came here, I think 2015.
Yes.
And he has been, he also has volunteered to be a peer mentor, and he came to my classes and shared his experiences with other master's students and gave them a lot of advice, and spent a lot of time in that social realm, which I really appreciated. Thank you.
Thank you. Always really. I don't want to cry in here, but that's so nice, so kind of you. That's so kind of you. Thank you so much.
For people online that couldn't see, Yiheng is crying right now. Okay, so if there's no more questions, we would like to congratulate Yiheng again for your great achievement and all the good luck for your future research.
Thank you so much. Thank you. Thank you so much. Have some lunch, please.
So Eric said, great job, Yiheng.
Oh, thank you so much. Thank you so much. Thank you so much. Sure.